We have prepared these solutions required for conducting this MTTS assay. Now let's proceed to see how we can start and set up this assay. ये स्लाइड आपको जो है इसमें लोअर पार्ट शोज दैट 96 वेल प्लेट का जो एक स्केच है दैट वी हैव available in our labs, and we use these plates to conduct to perform this test. We are going to start with these cells to be cultured in it, and then going to perform the further steps. चूँकि 96 वेल प्लेट है, इसकी वॉल्यूम लिमिटेशन है, सो द कल्चर मीडियम जिसमें हमने सेल्स लेने हैं, रिक्वायर्ड नंबर ऑफ सेल्स लेने हैं, उसकी भी लिमिटेशन आएगी. सो 100 माइक्रोलीटर पर वेल इज द रिक्वायर्ड वॉल्यूम ऑफ द culture medium containing the cell. We are going to pipe it in our 96 well plate. The per cell volume, well volume is going to be 100 microliter. Once we add the cells in this 96 well plate, we have to give it some time so that the cells can settle down, they can stick with the base of the well, and they can start normal growth cycle. It can be overnight incubation, it can be 4 to 6 hours incubation. That you have to optimize, that you have to decide for your specific study. The next step would be the setup of the assay. In this 100 microliter culture medium, we are going to add 10 microliter MTT solution. The solution which we prepared, and we ensured that this is the sterilized solution. We are going to add 10 microliter in each well, and ultimate concentration of that MTT is going to be 0.45 milligrams per milliliter. After the addition of MTT solution, we have to incubate this 96 well plate in dark, and that incubation time, again we have to decide, it should be in between 1 to 4 hours, 1 hour, 2 hour, 3 or 4 hours. It again depends on your experimental protocols, that how much time you are going to give this reaction to take place. During this incubation time, the tetrazolium is converted into formazan, the crystalline form. Now the next step is going to be the solubilization. For solubilization, we have the solution 2, the solubilization solution, which is acidic by nature. We are going to add 100 microliter of solubilization solution in each well. So the total volume now each well contains is going to be 210 microliter. This addition is going to be followed by an incubation or by a warming phase, that we are going to place that plate at 37 degrees centigrade for 10 to 30 minutes. This 10 to 30 minutes time period is sufficient enough to solubilize the formazan crystals and to develop the color, the purple color, and later on we can mix the formazan crystals by multiple pipettings. For that purpose, we do not need to go for extensive mixing using any vibrator or a shaker. We can simply take this multi-channel pipette, and pipetting up and down will ensure it to thoroughly mix the formazan crystals into the given volume of the solution. And now this is the time for recording the absorbance. The color intensity has been visible, has been seen changing in that particular wells of the 96 well plate. The absorbance can be read in the microplate reader, and the results can be seen as well as the optical density values can be obtained by a pre-programmed the optical density calculator. This slide only gives you an idea about the variations in the color intensity and the cell viability. If we look at the very first column and rows, like this one, the color intensity, and we compare it with this one, color intensity, and with this block where the yellow color prevails, that indicates that the viability of the cells in MTTS assay is seen to be decreasing from left towards right, like this. And by this means we can proceed further to calculate the number of viable cells which we are going to use for subsequent research experiment.